Okay, this is our last set of lectures, and we're going to be covering biotechnology. So really what we can do to manipulate DNA and how it can help, humans in general, but could also be used for lots of other things as well. So we're just going to scratch the surface with some of this technology. There's a lot more found in your book, and when you go into molecular and genetic biology, you'll get a lot more of this as well. All right, so the principle of being able to manipulate DNA goes to this principle of making DNA or taking DNA from different sources and combining it, really making recombinant DNA. And this is made possible by enzymes such as restriction endonucleases. And these are enzymes that recognize specific sites, specific patterns in DNA, and are able to cut or cleave at those sites. Okay, so you kind of see over here we have the EcoR1 site, which recognizes this specific sequence, and it will cut at that site. It's originally used by bacteria against viruses, so it recognizes specific sites of viruses, but we have since used it to manipulate other genomes. We also have restriction enzymes, okay, and these also cut DNA at specific sequences and can lead to what's called sticky ends. So basically, part of the DNA strand is left open to where if you have a sequence which matches that, it can stick together. Okay? So in order to do that, though, you need the matching sequence. So you could have a gene, and you could use restriction endonucleases or restriction enzymes to cut and create these sticky ends. And if you do it in two different pieces of DNA, you could get those two different pieces of DNA to stick together. And then DNA ligase is used to create the phosphodiester bond between those two sticky ends. Okay? This is the same enzyme which was used to join together the nicks in the Okazaki fragments of the lagging strand in DNA replication. So if you put all these things together, you can take DNA from two different sources and combine them. Now, there's a lot of other methods used to detect genes, usually by size or by charge. So one of those is called gel electrophoresis, which separates DNA fragments by size and charge through a gel. The gel is made of agarose or polyacrylamide, and it's submerged in a buffer that can carry a current. So when you put a current through this, you will create a charge, and then the pieces of the DNA which you may have in these different reactions or these different experiments which you have conducted are going to be attracted to the positive or negative end depending on their charge. And then the smaller fragments will move faster than the larger fragments, and so those will separate throughout the gel, and this is what it looks like down here. Oftentimes a fluorescent dye is used, or you can use non-fluorescent dyes as well. And that'll just help to visualize how big and how many fragments you've created, and so forth. Now, another way in which you can, join DNA, make more recombinant DNA, is a process called transformation, where you're inducing foreign DNA into a cell. This occurs in many species. E. coli does not naturally do it, but you can induce it by exposing E. coli to temperature shifts. Okay, and by shifting the temperature up and down, it will then transform its DNA, and part of it will go into different organisms. All right, and then we'll talk about another process similar to that later. All right, molecular cloning is creating an exact genetic copy. And this is done through molecular cloning, where you isolate a specific DNA sequence, usually one that creates a protein, some sort of protein that you want, right? Sometimes called gene cloning, and the most abundant organism we use for this is E. coli, which is naturally found in the gut of most organisms. The vector in this instance in molecular cloning is what carries the DNA in the host, and then it also can replicate in the host, so our host is generally E. coli, and each host vector system has particular use depending on what functional protein you want it to make. So some of the vectors that can be used are plasmids. These are small circular chromosomes, usually much smaller than a prokaryote chromosome, and they're used for cloning small pieces of DNA. Okay, so you may have this gene that you use restriction endonucleases to cut and stick a new gene, foreign DNA, in there, so now you're making recombinant DNA, and then inserting that plasmid within your E. coli or whatever bacteria or other organism you might be using, and then if it makes that protein, you can detect then if it was successful or not. So there's three components to a plasmid vector. You have an origin of replication, which allows it to be replicated similar to prokaryote chromosomes. You have a marker, something that allows it to be identified, and then you have sites on it which allow it to be cloned, so multiple cloning sites. Alright, so there are medical applications for this, including if you put a gene in, this is a common use for these applications, using a vector, using restriction endonucleases to insert a gene into E. coli. You can insert the gene for human insulin into E. coli, and then it will produce insulin. You can also use it for vaccines. The biggest problem is being able to purify the product. So it will make insulin, but also make all its other products as well. So being able to purify that insulin is a problem. Alright, so for making vaccines, one process is called making a subunit vaccine, and that's what this process is here. So what you do is genes that encode for part of a protein coat for a disease, let's say smallpox, you can take part of those genes for the protein coat, which is going to be part of the virus, splice it into a fragment, and then insert it into a new virus like the cowpox virus, which is harmless. So you take part of that gene, insert it into the cowpox virus, that you then inject into a person. The person is going to develop antibodies against that gene which you inserted in there, and now you have immunity. DNA vaccines are done a little bit differently because those are usually done with cellular processes and don't involve the immune response, the immune system. So gene therapy then is taking advantage of some of these methods to maybe delete genes which would be problematic, create these genetic disorders, and or inserting a gene necessary to correct for these disorders as well. So one example of this is severe combined immunodeficiency disease, which is a genetic disease. Some of the trials have shown success. So 15 children treated for this using gene therapy were successfully treated and they're still alive. However, there are many of them, 3 of them, which were treated and developed leukemia. So if you're manipulating genes, sometimes you can mess up the genetics of a person and cause, things like cancer to occur because you messed with the replication enzymes or whatever. And so then we'll talk about cancer later, but that's the generals of it. You can also use these in agricultural applications. So there's a plasmid called the tumor-inducing plasmid, or the TI plasmid, and it's used as a vector for plants in genetic engineering. And this is obtained from a bacteria which also uses this to infect its genes into plants, which normally infects broadleaf plants, so it can't work for non-broadleaf plants like corn and rice. But what it does is this bacteria will insert its plasmids into a plant, and then that plant will start creating this callus growth. It's kind of like a cancerous growth. Well, we have taken advantage of this and inserted genes of our own. And so one of the things you can do is you can insert genes for resistance to glyphosate, which is the principal ingredient found in Roundup. And so if these plants are resistant to Roundup, then you can spray your crops with Roundup and everything else will die except for your gene resistance plants. A similar thing has been done with an insecticide. So the Bt toxin from a bacteria, Bacillus thuringiensis, is something that it releases a toxin which keeps insects away. So if you put this gene in these plants, they're resistant to most insects. And this is the second most common genetically modified crop globally. A stacked crop is one that does both of these. So it has glyphosate resistance and Bt producing, which would be ideal because then you wouldn't need insecticides and you could spray it with Roundup and everything else would die. You wouldn't have to weed. All right, another use of this was inserting genes that you may want in a plant or you may want in a food. So golden rice is an example of this, where you put in a gene, a recombinant gene for beta-carotene, and put it into this rice. It made it a different color. It's kind of yellowish. And it couldn't be done by conventional breeding because the rice does not naturally produce the genes for beta-carotene. It doesn't have those enzymes. But you put this complex of genes in there which allows them to do it, and now they can produce beta-carotene. And now this rice can be provide a vitamin which would otherwise be unavailable. And whereas Roundup Ready plants, which have glyphosate resistance, are patented by the company that makes Roundup, these are do not have any commercial entanglement, so these are open for anybody to use. All right, another example of a genetic tool used in plants is called marker-assisted breeding. This is where you do classic plant breeding, so selecting for traits just artificially by breeding, and using molecular biology techniques to make a more hardy plant or resistant plant or whatever. Screening for it uses DNA fingerprinting, which we didn't talk about, what that is in your book. Basically finding the genes, inserting genes, finding which crops have these genes, and then selecting for those individuals and making them, making a seed crop from those. So genetically modified organisms are very abundant in the food which we eat, and this is a recent controversy, is making sure that we label these. Some health conscious people want these things labeled in our grocery store, similar to how organic crops are labeled. And the idea is that we don't know if genetically modifying our foods is going to create health problems for us down the line. Right now, there seems to be no evidence for it, but maybe maybe after eating foods like this for the next 50 years, it'll create problems in our health. We just don't know, and so that's why some people want it to be labeled and to act cautiously with these genetically modified organisms. All right, another technique we can use plants for is for creating pharmaceuticals. So if you can insert genes for creating specific, chemicals that can be used in our pharmaceutical applications, that would be a great way to produce them. In 1990, the human serum albumin, so this is a very important part of our blood, plasma protein, was genetically engineered in tobacco and potato plants, and there are other types of biopharmaceuticals also being produced, such as a recombinant subunit vaccine against Norwalk and rabies viruses, and a recombinant monoclonal antibody against tooth decay causing bacteria, which would be nice. Transgenic animals have not been as abundant and not as successful, but there is some research going into making something similar to biofarming, where you can have pharmaceuticals naturally produced in milk, and so you could just drink this milk, which has these cows have genes within them to create, I don't know, whatever, Tylenol or whatever you want them to produce. All right, so that's those are genetic, biogenetic tools and techniques. Last section we're going to talk about is sections of the cell cycle and then cancer. So we've talked about the cell cycle, the G1S, G2 phase, and the M phase, and then you have cytokinesis. The control of this, so what makes it go from one phase to another, has the current view integrates two concepts. Okay, the first one is that the cell has irreversible points, and two of these are where the genetic material is replicated. Okay, so that's going to be right here before the S phase, and the second one is right here before the separation of the sister chromatids, so right before anaphase. The second one is that the cell cycle can be put on hold at these specific points called checkpoints. So we've got 3 checkpoints which are actively being studied. The first one is the G1S checkpoint, where the cell decides that, okay, it is done doing whatever it needs to in the G1 phase. It's fully functional, part of the cell, and is now going to go into preparing to divide. The second then checkpoint is the G2M checkpoint, okay, where it's basically ready to go through mitosis. And then the last one is the late metaphase spindle checkpoint, where the chromosomes attach to the spindle and they're getting ready to go to anaphase. Now, what controls these are cyclin-dependent kinases, or Cdks. And these are enzymes that phosphorylate or add a phosphorus to activate or inactivate proteins. So if you phosphorylate, you are adding a phosphate group to the amino acids on the chain of the protein. So cyclin-dependent kinases partner with different cyclins, and cyclins are proteins that are made at different points in the cell cycle. And then these CDKs can be activated or inactivated by phosphorylating. So you may put a phosphorus on this site, which would turn it on, and a phosphorus on this site would turn it off, or some combination of the two. These are also called mitosis promoting factors, the combination of cyclin and CDK. Okay, so you have an interaction of two proteins creating this. Damage to DNA will cause phosphorylation of mitosis promoting factors to its inactive form, and thus mitosis will be halted and will prevent the continuation of a damaged DNA cell to continue. So we're going to look at yeast, and we're going to look at other eukaryotes. In yeast, the cyclin-dependent kinases have a few different checkpoints. The G1S CDK checkpoint, so this is the first checkpoint here, which is a combination of the CDC2 CDK and the G1 cyclin. And this increases the enzyme's activity necessary for replication. Okay, then you have the MPF checkpoint, which is the G2/M checkpoint, where you have the CDC2 and the mitotic cyclin combining. And then you have the anaphase promoting complex, or the spindle checkpoint. Alright, in multicellular eukaryotes, it's similar, except we have two different CDKs. We have CDK2 at the G1S checkpoint, and then you have CDK1 at the MPF checkpoint, and then anaphase promoting complex. Alright, some other things that affect cell growth are growth factors, and these act by triggering intracellular signaling systems. So growth factors attach to a receptor. That receptor causes different parts of the cell to grow, okay, and divide. The most common one, or first one discovered, was the platelet-derived growth factor. And these can override cellular controls that would otherwise inhibit cell division. So these controls are keeping cell division from going on. You get growth factor in there, and it decreases the inhibitions and allow the cell to divide. Now, cancer occurs when you have unrestrained, uncontrolled growth of cells. Basically, you have mitosis occurring at an unnatural, hyper-elevated rate in a specific tissue or cell. So you must have some sort of failure of the cell cycle for cancer to occur. There are two kinds of genes that can disturb the cell cycle when they are mutated. One is tumor-suppressor genes, and one are proto-oncogenes. So we'll look at tumor-suppressor genes, one of them, and that is a gene called P53, which plays a key role in that first checkpoint, the G1 checkpoint. P53, what it does is it monitors the integrity of the DNA. If DNA is damaged, then cell division is halted and the repair enzymes are stimulated. If DNA damage is irreparable, P53 turns on and says, all right, we're going to kill this cell so it doesn't continue to affect further cells down the line. And so it prevents the development or continuation of mutated cells continuing. P53 is absent or damaged in many cancerous cells. So when they looked at different cancerous cells and checked the different genes of them, usually P53 is not there. All right, proto-oncogenes are genes that are normal, but when there are malfunctions in these genes, they become cancerous. So a couple of them have been identified, BRCA1 and BRCA2. And some of them encode receptors for growth factors, like we showed before. If the receptor is mutated in the on position, the cell will no longer depend on the growth factor, and it will grow and divide without the stimulation. And only one copy of one of these genes is needed for a cancerous cell to be produced, or the uncontrolled division to take place. So I have a picture of Angelina Jolie here because she had a couple of genes which I mentioned before, BRCA1 and 2, which are connected to ovarian and breast cancer, and so she had her ovaries and breasts removed within the past few years. And also kind of did it publicly to say, all right, you should go out if you're a woman and have these be tested for these, and if you have them, consider having your ovaries and breasts removed. Also, just a few years ago, there was companies who wanted to patent these specific genes. Went all the way to the Supreme Court. It said that no, you cannot patent a gene. Okay, so it's part of the human body. It's not something that you can have, control over as a private company.